So we'd like to call upon the next chairperson, Dr. Govindappa D, who is the consultant physician at Govind's Medical and Diabetes Care, Bengaluru. We welcome you on stage, doctor. Oh God. Who else? Sir. Just wait. I would like to invite Dr. Vitul K. Gupta to Shashank Joshi.
So I invite great academician, leader, inspirational figure, Dr. Shashank Joshi, to speak on rather important topic presently, the metabolic revolution, GLP-1 RAs for holistic care in type 2 diabetes. Definitely, holistic care means each and every aspect of diabetes, including reversal of type 2 diabetes. I invite Dr. Shashank Joshi. He doesn't need any introduction. The person who doesn't know him has no connection with academics. Thank you.
So Vitul, thank you so much. Thank you, Srinivas. Actually, this is a sponsored talk by Abbott. They have partnered with Novo Nordisk to launch semaglutide, which is, there are two wars happening. One is war against diabetes, and second is war in Iran and Israel. And now missiles are being fired on price cuts, etc. And this is a very hot topic. So when we cross the millennium, the children who are born, I am alluding to what Dr. Das was saying, they were called as millennials. Then you had the next generation, which were the Gen Zs. Then they were called alphas. The children who will be born today, they should not get adolescent obesity, and therefore they will be called as a GLP-1 generation. And I'm sure you must have heard of Ozempic babies and all. And now Novo has partnered with the Ozempic team to get a, with Abbott called Extensior. So I'm going to talk a little bit on GLP-1. So those are my duality and conflict of interest for this talk. So metabolic obesity is very prevalent in India. We are thin fat Indians. We have a lot of abdominal fat, insulin resistance, a typical dyslipidemia. Even at a normal BMI, we have something called as mono. And I'm a part of the study with Professor Mohan from the ICMR INDAP cohort. And there are two types of obesity: metabolically obese, non-obese, and metabolically obese. 28% is metabolically obese. But 43% of people living in India, and this is from the ICMR 23 INDAP study, are mono. Metabolically obese, non-obese. So my first take-home message to everybody is, we have a large population with normal BMI, which has abdominal fat, insulin resistance, and a dyslipidemia, with increased risk for type 2 diabetes, cardiovascular disease, and chronic kidney disease. And 75% of type 2 diabetes have at least one microvascular complication. And duration of diabetes is recognized as the main risk factor for developing microvascular disease, but unfortunately we diagnose them because more than 50% of people don't know they have diabetes. First time they know they have diabetes is when they get a complication. GLP-1 agents are revolutionary and game changers because they're anti-diabetic agents beyond pancreas. Today is the day when it is getting off patent, but they typically switch on the beta cell to make insulin, switch off the alpha cells to reduce glucagon. They reduce the hepatic glucose diarrhea, that's why they control the glucose, but they are not just diabetes agents. They reduce appetite, they enhance the incretin effect via the vagal nerve, and they improve the glucose uptake in the peripheral tissues. So they address every core defect of type 2 diabetes, And we clearly recognize that the recombinant DNA injection semaglutide has its approval in India as an adjunct to diet and exercise. Its cardiovascular risk reduction for major MACE, for cardiovascular death, and for CKD. So those are the 3 approvals which are in place for that particular originator. There are more than robust clinical development has undergone in a semaglutide program, and Dr. Srinivas Murthy, who is the organizer of this meeting, has been a part of all those Novo trials. In fact, his center, you should put your hands together for him, is the US FDA and EMA Center of Excellence, and he's a Novo Nordisk Center of Excellence. So people who have to do trials even in Europe come to his center to visit it. So he has done all the 7 trials, which you can see here, Sustain, 1, 2, IP, Sustain. So there are more than 1,246 plus patients from the Indian origin, and almost 300 come from his center in Bengaluru, and more than 17 trials with 20,000 population has been studied on this molecule. The A1C reduction of this molecule is 1.6% versus gliptins and 1.5% versus SGLT2 inhibitors. They cause weight loss, they have better patient compliance once a week, cardio protection, kidney protection, and clearly head-to-head they also score over insulin, that is glargine or aspart. And clearly there is no hypoglycemia, once a week injection, better patient compliance. So as an anti-diabetic agent, don't think them as weight loss agents. As an anti-diabetic agent, it's an excellent agent. Our cardiology colleagues can use it, nephrology colleagues can use it, peripheral vascular disease surgeons can use it. It's an excellent agent because it's an agent with outcomes. See, we are in the evidence-based era and we have to look at outcomes. Sustain 6 had people with either history of CV disease or CKD and clearly showed 26% reduction in macrovascular or MACE outcomes. Nephropathy, 36% reduction. So clearly with high cardiovascular risk, you can see that Dr. Suresh is here, he's a cardiologist, he will also agree that from a cardiac standpoint, it has a huge outcome. STRIDE trial, it's the only trial with systemic symptomatic peripheral artery disease. Semaglutide has shown to improve the walking distance, making the patient claudication free. And people with peripheral artery disease and type 2 diabetes, it's huge. The FLOW trial has shown CKD progression reduction. And it can be also combined with SGLT2 inhibitors with a lot of synergy for lowering all-cause mortality, cardiovascular, renal, and glycemic mortality. So therefore, in the standards of care in 2026, it's a first-line therapy. If you look at a February NICE guidelines, it's a first-line therapy. If an injectable therapy is needed to control A1C, it should be considered before insulin. And it's a very high efficacy drug, both for glucose and weight control. But it's a peptide. And a lot of people have launched a lot of peptides today, and some of them are biologic, some of them are synthetic. The rDNA is the one which we have studied for 104 years. And that's something unique. That's what Novo and Lilly are popular for. And therefore, why should we as doctors care about the process? Process defines the product. Because there is a gene of interest identified and cloned, the vector is inserted, it's transmitted through a cell culture, there's downstream processing, a pure substance is made, and we formulate it through a proper cold chain. So that's the rDNA product, which is what the innovators have made. And of course, we have a plethora of generics which will be launched, which are all made by chemical synthesis. So the bacteria, yeast, all of them have greater purity, stability, scalability, environmental sustainability, while the peptide, synthetic peptides which are now being manufactured and made are basically assembly of amino acids in a solid resin with organic solvents, and basically it allows production of complex peptides but also generates waste and introduces impurities in the substance. So we need to understand that this is, in the rDNA space, we had biosimilars. So what is being launched as generics are actually not biosimilars. They are synthetic copies. They are not even generics because small molecules are generics. So we need to distinguish all the three. Biosimilars are from rDNA. Generics are chemical peptides, so paracetamol to paracetamol, for example. And these are synthetic copies. A synthetic copy is made through a cleavage from a resin and is purified. That's how it is made. And there is data around that. And therefore, they are not identical. They are not very clearly identifiable. So, original biologic can either have a follow-on biologic or a follow-on chemical drug. We don't know much about it. But remember, a complex biologic is not simple as a photocopy. Because as I told you, aspirin is a small molecule. It's very easy to replicate. Insulin, growth hormone, clotting factors are large molecules. And then there are smaller proteins like semaglutide, which actually are manufactured by both processes. So different manufacturing processes result in different product characteristics and impurities. And therefore, in the entire biologic, it's a very well recognized chain. And the peptide structure, the synthesis, the formulation, the degradation of a quality peptide drug is very well established. So a chemical synthesis of a complex peptide may alter function of the product and clinical consequences. That's why we need to do clinical studies. And therefore, if you look at the process of synthetic or compounded semaglutide, there is a study published by Sabi et al. And Hack et al. Where they clearly published in 2020 and 2024 in US, Europe and Asia and North America, South America, Europe, Asia and Oceania, that there was synthetic liraglutide versus recombinant liraglutide and synthetic semaglutide. And they studied the samples. They did chromatography, they did spectroscopy, and they did safety assays. And they were able to identify chromatography and mass spectroscopy of more than 100 new impurities, amino acid deletions and additions, unlisted excipients and polymers, a contamination which was seen, and therefore lack of GLP-1. And also there was a high recognition of solvents, buffers, nitrates, trace elements, and ion analysis. And on structural spectroscopy, they found that there was an altered higher order folding because proteins have to be folded. And also we remember that chromium picolinate interferes the masking of semaglutide signaling. So incorrect folding compromises the biological functioning and efficacy. The fibrillation time is shorter because when there is rapid fibrillation, there is a loss of active drug. So the safety and immunogenicity could be a problem. And then when there is a drastically lower pH, the dissolution may be a problem and there could be therapeutic failure. And the MHC protein on new isotopes could be difference. So fibrillation is a big problem with a synthetic peptide. It's a tangled mess and therefore you might not get the same immunogenicity. So obviously the regulatory framework, how synthetic copies are evaluated against original is actually very robust. And it's a very extensive approval pathway and therefore we need to do a lot of phase 1, phase 2, large population phase 3 and then proper basis. Of course now they have given this. So clearly there is a gap with synthetic versions. Minimal phase 3 evidence, no long-term outcome data, limited label indications, limited or no real-world data, and no overall data. So obviously the innovator is rDNA, extensive RCTs and real-world data. It's global and Indian, consistent cold chain, and has more than 40 million patient years. So clearly we have a challenge. So we have rDNA available with us. It's also reasonably economical. It has gone through this entire process of characterization, which is very robust and not shortcutted, while the synthetic semaglutide has gone through a shortcut and we don't know its efficacy. We'll have to study it once it is available. We don't have long-term cardiovascular data, renal data or PAD outcome data on that molecule. But having said that, semaglutide is here to stay. You know, that was just a point because this is a point for Srinivas Murthy and the innovator and they have funded his meeting. So I didn't have a choice but to talk of all this. But there was some science around it. So I didn't want to totally dilute the science as well. That we should be mindful that we are looking at two different compounds. One is synthetic peptides through a generic route, which need evaluation and more science on them. And we have the rDNA product available with us. But the molecule semaglutide is very unique. 10% weight loss, up to 3% A1C reduction, improvement in claudication distance and walking distance, 20% reduction in all-cause mortality, 24% reduction in kidney disease events, and 26% in MACE. So an extraordinary molecule on triangle of care. Whether it is the liver fat, whether it is the kidney fat, or any other fat anywhere else in the body, it's a very robust molecule. And GLP-1 receptor agents are here to stay. Very, very clearly we have now in pharmaceutical management of type 2 diabetes and people living with type 2 diabetes, we look beyond glucose. We are looking at outcome-based medicine, whether it is cardiovascular outcomes, renal outcomes, liver outcomes, knee osteoarthritis outcomes, sleep apnea outcomes. We have a very, very good and robust agent which is available. We are managing weight and glucose both. And therefore, in the landscape of metabolic disease management, we have an agent which is available. Thank you so much for a patient hearing.
Thank you, Dr. Shashank, for making this valid point. Not for the even drug, whatever we eat, we should know about the quality and how it is produced. So that is there. Definitely, semaglutide is a great molecule, but we should know what we are eating, how it is being produced. Thank you very much. Any comment from my co-chair?
Sir, we excellently have highlighted the difference between the biosimilars and synthetic peptide, semaglutide, and also the role of this GLP agonist in the metabolic evolution. You know, as we know, the diabetic modern therapy is not just glucose control, not just HbA1c control. We have to protect different organs. It's a metabolic revolution with the availability of GLP-1 agonist and SGLT. These two drugs have certainly produced metabolic revolution in the field of diabetic care. We have got these drugs to protect kidney, heart, lung, and metabolic control, and to a certain extent in mental stress reduction also. You have very nicely highlighted, sir. Thank you very much for your lucid talk on GLP. That is why the topic has been given as holistic approach.
Absolutely, I fully agree with you. These are disease-modifying drugs, and these are anti-complication drugs. If you use these drugs early and hit early in a type 2 diabetic person, you will be able to keep them free from complications. And if they have pre-existing complications, and if you use them efficiently, you will be able to keep them and make them live longer.
Thank you. For constrained time, we are closing the session. And Dr. Shashank is always available to answer any query, anytime, anywhere. Thank you.
There is a saying, to make a mistake is human and to forgive is divine. So my apologies to you, doctor.